share this one. Can everybody see the wonderful biochemistry?
Yep.
Okay. Okay, you guys. We're going to do our best to try and get through these. Honestly, a lot of this kind of general stuff, like these kind of questions are going to be almost, a good handful should be almost word for word what you'll see on the exam. So even like what I've been noticing is he seems to be picking from a lot of like the LINUS stuff even more than he used to. So yeah, we'll run through it. Okay. So
Can somebody send me the questions?
I'll add
Yeah,
you
I
to the
can just-
Google Drive I have them in, JC.
Thank you, thank you.
If you will send me either in GroupMe or something, send me your. Do you have a Gmail?
Yeah, I'll send it to
Okay, send me your Gmail and then I'll add you to that.
you. Perfect, thank you.
Mm-hmm.
And I just dropped them into this chat window as well. So. Okay. All glycerol-containing phospholipids are synthesized from which of the following? This one you're going to see a lot in a lot of different answers. Answer is phosphatidic acid. If we get to there, we'll talk about we'll look at what cardiolipids and that kind of stuff. Mevalonate, we should recognize this from last week's tutoring session. Does anybody remember what mevalonate is? It's okay if you don't. You can tell me that you don't.
What's that, Drake?
Thank you, JC.
Like it sounds vaguely familiar.
Mevalonate, if you remember, it's that 3-carbon molecule that's produced when we break up an odd chain fatty acid. Does that ring a bell?
Yes.
Yeah. And then we'll also, from there, we'll also look at mevalonate's also going to be important in certain situations when we talk about synthesizing or making fatty acid chains. Okay, so we'll get to that a little bit later on. Okay. And hey, look, it's like literally the exact same question. Repeat it again. All glycerol-containing phospholipids are synthesized from which of the following, you guys?
Phosphatidic acid.
Yeah, you're so smart. It's crazy. Okay, so the biosynthesis of palmitic acid does not use which of the following? We have biotin, bicarbonate, NAD+, or acetyl-CoA. Any guesses before we go to the book?
NAD+.
Yeah, NAD+ is going to be the correct answer, that we do not use NAD+. Let me highlight. Jumping over to the book. So some of the requirements of fatty acid synthesis right here. Some of the stuff that we're going to be needing. Acetyl-CoA. This is going to come back later several times. Acetyl-CoA is our very first source of carbon. Okay, so the first carbon that we're going to add into a fatty acid chain is going to come from acetyl-CoA. Okay, we're also going to be using NADPH. Again, because we are forming bonds, we're building bonds, we need to use ATP. HCO3 is bicarbonate. That's going to be the source of our second carbon. Those are the big ones we want to remember. We want to remember where we're getting these carbons from, okay? We have biotin. Remember, biotin is one of our B vitamins. It's called a vitamin. What else do we call vitamins? Remember, our minerals are cofactors. What are vitamins?
Coenzymes.
Yeah, our vitamins are coenzymes. Biotin specifically, what is biotin? What vitamin is biotin?
B12.
Pickle. Ball. Centuries would be 12. 9.
Tall.
6.
Nudis. Played. Pickle. Ball. 4. Centuries. 9 and 12. Always the last two. Thiamine, we have 1, 2, 3, 5, 6, 7, 9, and 12. So biotin is our B7. And then we have Mn, manganese is our Mn, right? Mn is manganese, or is that because Mg is magnesium?
Yeah, Mn is manganese.
And then fatty acid biosynthesis involves condensation of successive 2 carbon units, which are malonyl CoA. Malonyl CoA is an intermediate of the fatty acid biosynthesis and serves as the donor of 2 carbon units. The third carbon is eliminated as carbon dioxide. It should be a nice little picture here. So we have beta oxidation. This is important to understand. Beta oxidation is going to occur inside of the mitochondria, inside of the mitochondrial matrix. The whole goal of that is to produce a bunch of acetyl-CoA to put into the Krebs cycle, to put into other stuff to give us energy. But unlike how we talked about kind of, glycolysis, gluconeogenesis, a lot of gluconeogenesis was just kind of like a reversal of glycolysis. This, when we're talking about fats, they're two completely separate processes with like separate happenings, okay? I'm gonna make sure that's really, really clear that the beta oxidation is in the mitochondria, and biosynthesis, so building of them, actually happens within the cytoplasm. Okay, so make sure that you understand those are two separate things, very separate processes. They're not like happening in the same area. Okay, we will continue to come back to this a little bit as we keep going through stuff. So the biotin, bicarbonate, remember bicarbonate is HCO3, and we'll be using bicarbonate. Acetyl-CoA, is that the first or the second carbon that gets donated?
Second.
Yes.
Acetyl-CoA is the first carbon. That's why I wanted to make sure. This is the first carbon donated. Bicarbonate is going to be the second carbon donated. That's going to come back up several times as we keep going through these questions, okay? By what mechanism does palmitoyl-CoA regulate fatty acid biosynthesis? It regulates it by negative feedback regulation. It's converted to stearic acid, and then stearic acid transcriptionally regulates fatty acid biosynthesis. As we're going to start reading these couple words, just make sure we understand what's going on. Transcriptionally regulates. Does anybody have any idea what that means?
Nope.
Kind of what that's referring to? So remember in Biochem 1, when we talked about nuclear receptors.
Mm-hmm.
Right? So nuclear receptors, they go right through the membrane, they go right into the nucleus, and then they can bind down in there to stop actual like genes and proteins and stuff from being expressed. That's what we talk about transcriptional regulation. Okay? If it goes in and blocks certain receptors and stuff, in order to prevent it from doing that kind of thing. About that a lot, but just make so that way you kind of have that in your toolbox of knowledge. Okay. We good?
Yep.
Yep.
Okay, make sure it's still recording. My internet lagged for a second. Okay. Next answer, it activates the polymerization of the acetyl-CoA carboxylase and thereby increases palmitic acid biosynthesis. We're gonna look. We'll look at the book in a sec, but the palmitoyl-CoA is in most situations whenever you're gonna guess about something regulating something else. 9 times out of 10, we're looking at like negative feedback loop kind of stuff, right? When let's talk about negative feedback. What does negative feedback loop mean?
Basically, like we have enough of it, so it tells it to stop making it.
Exactly. So we continue down the line. We're making stuff. We get to an end product, and once we have enough of that end product, it kind of floats back and turns off the genes and the enzymes and stuff that are before, in order to turn them off to stop producing product once we have enough of it. Most of the time, we're going to see negative feedback regulation. There's just a handful of things that do like feed forward, or positive feedback regulation is what we call it. Okay. By phosphorylating, I just say that increasing palmitic acid biosynthesis, so rarely are we going to see feed forward stuff. By phosphorylating the rate regulatory enzyme in the fatty acid biosynthetic pathway. So in this case, palmitoyl-CoA is going to do exactly that. It's going to have negative feedback regulation. Let's jump in the book really quick and see if we can find exactly where the palmitoyl-CoA is and find that actual reaction. Might take me a second to actually find it. Couple quick things of review as we go through. We're going to be using NADPH in the production of our fatty acids. Always remember that we get those from our pentose phosphate pathway. That was at the very end of Biochem 1. The transport of acetyl-CoA to cytosol. This is another thing that we'll look at where, once we make a bunch of once we have acetyl-CoA, if we have extra acetyl-CoA, we need to pump it out. Do that. Give me 1 sec. Had to say goodnight. Transport of acetyl-CoA. This is one you're gonna want to spend a little bit of time and look at that whole process. We'll see if it comes up in a couple of questions that we can go over it a little bit deeper. This is going to be another one that you're probably going to want to draw similar to like the malate aspartate shuttle and all of those last trimester. Pretty similar type process where, we have citrate being able to come through and it gets converted back into pyruvate. Pyruvate's able to transport back across into the matrix, just like it would after glycolysis. And then we can bring that acetyl-CoA back and forth to get the acetyl-CoA into the cytosol to produce the fatty acid chains that we need to. Let me see if I can find the palmitoyl-CoA. There we go. So we'll go a little bit deeper kind of into these exact steps as we continue to go through here. But we start off with that acetyl-CoA. So this is just specifically the regulation of acetyl-CoA carboxylase. Acetyl-CoA carboxylase is going to be kind of one of our starters of fatty acid synthesis. So if we have a whole bunch of citrate, it's going to activate it. If we have insulin being pumped out, remember what's the job of insulin? What does insulin do?
Decreases blood sugar.
Yeah, and where does it pull that sugar into?
The liver.
Into the liver, into cells, just into cells in general, right? So
Yeah.
if we have more insulin going like activating, it's because we have lots of extra sugar. If we have extra sugar, we're running through that sugar, trying to break it down or storing it or whatever. Once our stores are full, we're going to keep breaking it down. Then we're going to have extra end products. We're going to have extra, doing glycolysis, producing pyruvate. That pyruvate is going to get turned into acetyl-CoA. Because we have a bunch of extra of it, then we know we're going to be like, okay, we're going to have to store some of this as fat. And some of the things that we're going to be inhibited, palmitoyl-CoA does the feedback inhibition, that negative feedback. Also, we can get phosphorylation of the acetyl-CoA carboxylase through CAMP-dependent pathways. Remember CAMP-dependent pathways? Those are our G protein coupled stuff. Remember those processes a little bit? So like glucagon, epinephrine, norepinephrine, when we have those guys, that's going to inhibit the acetyl-CoA carboxylase. Why do you think these three hormones specifically would inhibit the formation of free fatty acid, or inhibit the formation of fatty acids, to synthesize fatty acids to store them?
No idea.
Because it's telling the body that it needs energy, so instead of saving it for later, it cuts off that process so that energy can be used now.
Exactly, because our epinephrine, norepinephrine, these are our sympathetic hormones. Our fight or flight hormones are adrenaline. Epinephrine is known as adrenaline. Norepinephrine is known as noradrenaline. Both of them, adrenaline, they do similar jobs. Okay? They tell the whole body to kick it up a notch, breathe more, dilate the pupils, like be alert, like get up and go, start pumping the heart faster, get the blood running and moving. Like that's going to be our sympathetic nervous system. So in that situation, we need a lot of available energy to go and to do. And then glucagon, the opposite of insulin, right, is saying that our blood sugar is too low. We need some sugar into the blood. We need to raise it up. And so there's no reason we're going to be releasing that hormone in like a starvation state, in a non-fed state, as Dr. Sarkar likes to put it. And so we need our blood sugar to be going up. So there's no reason for us to be storing fat during that time when we need our blood sugar to go up. Makes sense?
Yeah.
Okay. Next question. CDP diglyceride is not involved in the biosynthesis of which of the following? We have phosphatidylcholine, phosphatidylserine, phosphatidylphanolamine, and sphingomyelin. This is where you kind of get like super specific, and I really don't like these questions because honestly a lot of it is just kind of raw memorization type stuff, and there are like some charts and stuff down a little bit further. That's the elongation. I'm pretty sure it's down here in this chapter. Yeah, here we go. Other glycerophospholipids and sphingolipids. So like all eukaryotes, mammals synthesis. Sorry, let me fix this real quick. It bothers me a lot. This like freaks out for some reason. There it goes. I just really messed with my eyes. Okay. Like all eukaryotes, mammals also synthesize glycerophospholipids from CDP diacylglycerol or diacylglycerol. The diacylglycerol is going to be broken down a little bit into the diacylglyceride as well. Those two are going to be connected. I don't know if it's the exact same thing or if it's just similar, but we have here this whole little kind of chart, and if we look through the chart, our glycerophospholipids are all of those phosphatidyl guys right there, whereas our sphingomyelin is one of the sphingolipids kind of on the other side. So CDP diglyceride is one of the glycerolipids, one of the glycerophospholipids specifically, a little bit different than our sphingomyelin. A lot of these questions, my recommendation is honestly just like throw these into a Quizlet if you can and see them enough times to just recognize them. He's gonna pull most likely like directly from these types of like from those specific questions, because sitting and trying to memorize this whole thing kind of sucks. You can kind of in general look at, you know, the patterns, like if it's a phosphatidyl group, bunch it all into that group, and then like if it's a sphingo, put it in the one specific spot, and then like kind of whatever's left over, throw into like the other two categories. But that's the way that I would approach that, kind of, because it's a lot. Put you back there. There. Next question. And again, at a point, you guys have questions, comments, things are confusing, stop me, let me know, and we can take a little bit more time walking through stuff. During fatty acid biosynthesis, which of the following enzymes releases palmitoyl-CoA from the acyl carrier protein into the cytosol? Again, this is going to be kind of part of the whole process. So we want to look at...
It can be like a thiolase.
Yeah, it is going to be the the
Thioesterase.
Yeah, the thioesterase. Let's find the I think it has the pattern written here as well. So we have, and we've talked about this a little bit before as well. We get activation. We have the fatty acid synthase primes the reaction, which should also, primes the reaction when we talk about beta oxidation as well. So fatty acid synthase doing its job. Reaction 2, Let me double check because it says the exact same thing. I want to make sure that I don't miss something. We missed the whole. Am I crazy or did we miss a step? This says second step. Where is first step? Here we go. First step. Production of mannonyl-CoA is the initial and rate controlling step in the fatty acid biosynthesis. So that is going to be something that we want to remember because we always want to remember the rate controlling steps. I'm going to say it again. Production of mannonyl-CoA is the initial and rate controlling step in the fatty acid biosynthesis. It is catalyzed by the acetyl-CoA carboxylase in 2 steps. And that is the rate limiting enzyme. That means where it's going to be important and we're going to focus on it. So taking acetyl-CoA, converting it into malonyl-CoA. We're going to use ATP, we use our biotin, we use carbon dioxide, all of that kind of stuff included right in there. This is just going to be kind of repeat, like more, a little bit more in depth kind of stuff of that. I don't want to jump too much into that. I just want to give us the basal layer. Our second step, the fatty acid synthesis from acyl-CoA and malonyl-CoA occurs by successive cycles of 6 enzymatic reactions. This is where we're going to want to, again, just like we did in beta oxidation. If we think back to beta oxidation, do we remember what our order of steps were? What 4 reactions we had in order for beta oxidation?
That's the one that had it was like the dehydrogenase, or yeah. Words are hard.
Yep. D hot, where the O is actually
Yeah.
a D.
Yes.
So we had this is beta oxidation. Just want to review this. Beta oxidation, dehydrogenase. Specifically, the first dehydrogenase. Whenever we dehydrogen something, we need something to hold on to that hydrogen. Do you remember what that first hydrogen, or what that first hydrogen is given to?
FAD?
Yeah, to an FAD. And then we have an H. What was the H stand for?
I don't remember.
Hydration. Hydration, so we're adding water back into things. And then we have another dehydrogenase. That one's for NAD. And our last step is a thiolase for beta oxidation. Cool, just a quick review of that. Keep it fresh in the brain as we get close to test time. So now we're going to switch gears back to the synthesis side of things. There's going to be 6 enzymatic reactions, 6 steps that we're going to be looking at, okay? This is what happens before those 6 steps. So we're going to look at that again. So activation of the fatty acid synthase by attaching an acetyl group to ACP, or the priming reactions. That acetyl group is first attached to the ACP to form acetyl ACP. And then in the next step, the acetyl group is transferred to the enzyme portion of the FAS. FAS is our fatty acid synthase. ACP is our acetyl-CoA carboxylase. I believe ACP. We'll go with that until proven wrong otherwise.
There it is right there. Scroll right back up just a little bit.
Acyl carrier protein. Thank you. Including an acyl carrier protein. Thank you. Second step, we're still activating the fatty acid synthase. We attach a malonyl group to that ACP. And then we're going to continue on down. Reaction 3 is a condensation reaction. Then we reduce, dehydrate, reduce, and that's what it says, I guess, our sixth step. I want to check, because there's supposed to be... I don't like the way this is all worded. 6 enzymatic reactions. It doesn't tell us what these are specifically called. So we'll go like this. We'll say priming, attaching. Sorry, my mouse doesn't work very well on this laptop. Condensing, reducing, dehydrating. Come on. These school laptops are not the way. And then reduction. So priming, activating or attaching, dehydrate, condensation, PAC, PAC, RDR. Priming, attaching, condensing, reducing, dehydrate, reduce again. We're going to repeat those to do all the stuff that we want to do. Which, geez, the original question that we were trying to get to is that during the fatty acid biosynthesis, which of the following enzymes releases palmitoyl-CoA from the acyl carrier protein into the cytosol? Within this mess, is that answer to that? Unfortunately, I can't go deep dive into this just with the amount of time that we have. So it's kind of buckle down, and I have this whole thing recorded on my YouTube video. So go through it, watch it, write down the steps. I know test is coming up quick, so if you can find time to do that ASAP, it's probably gonna be the good. Or honestly, if you're feeling really unprepared right now, best bet is probably going to be just throw all these into Quizlets and grind these questions as much as you can. So whichever stage you're currently in, do with that information as you will. Okay. Enzymes from which of the following pathways are utilized for fatty acid synthesis? So if we look at this citric acid cycle, glycolysis, pentose phosphate pathway. We know that from the pentose phosphate pathway, we're getting that NADHP, the citric glycolysis, right? We're looking at when we looked at the transport of the acetyl-CoA, part of that was turning things back into pyruvate to get it back across. That's with glycolysis. And then same thing with that citric acid cycle, converting things into acetyl-CoA inside of that mitochondria, all that kind of stuff, getting it out of there. Citric acid cycle, so our answer is all of the above. We're going to try and go a little quick because there's a lot of questions here. We'll see how many we can get through. Fatty acid biosynthesis differs from beta oxidation of odd chain fatty acid in its use of NADPH, involvement of citrate cleavage enzymes, all of the above are subcellular location. So immediately we look at with beta oxidation. Beta oxidation, we don't use NADP plus, right, are technically a biosynthesis. So we use the NADPH, so kind of close enough. Subcellular location, those are different because beta oxidation is in the mitochondria. The beta biosynthesis of fatty acids takes place in the cytosol. And then the citrate cleavage enzymes, again, those we're using as part of the transport. So if it's at least 2 of them, it's got to be all of them. How many molecules of acetyl-CoA and NADPH are required to synthesize 1 molecule of palmitic acid? This is going to be similar to kind of like the loops of everything. So we look, palmitic acid, if we think and remember palmitic acid, how many, does anyone remember how many carbons a palmitic acid contains?
Isn't it a 16 carbon fatty acid?
Good, yeah, 16 carbons. So if we have 16 carbons and every acetyl-CoA has 2 carbons, how many acetyl-CoA are we going to need?
8?
Yeah, so immediately we can cross out 1, bum. Actually, all of them, because the only one that says 8 is A. We need 8 molecules of acetyl-CoA, and then we will also need 14 molecules of NADPH. When you go over the specific step-by-steps, when you review those, kind of look at that, where the NADPH is being used up, okay? Kind of the goal here, if everybody has like a little notepad or something on the side, take note of the things that I mentioned to go back and look at, just that way you can find those. If you don't, it's something that you maybe don't feel confident right now. Identify the monounsaturated fatty acid from all of the following. This is just, again, it's in the book. There's a little bit of a list. He's going to give you kind of those, a chunk of those. Palmitoleate is the monounsaturated fatty acid. He'll give you a couple of like common mono and polyunsaturated fats that he wants you to know within the material. If malonyl-CoA is synthesized from 16 CO2 and unlabeled acetyl-CoA, and the labeled malonate is then used for fatty acid synthesis, the final product will have radioactive carbon in which of the following? So there's some biochemistry terminology in this. Labeled malonate. Labeled, which honestly, if I didn't, like in this specific context, otherwise I wouldn't understand what this is asking. Labeled means radioactive, okay? So we have.
What was the name of the starting carbon or the starting molecule that we work with? Do we have to start with one thing and then we add on the acetyl-CoAs afterwards, right? Is the malonyl the one that we start with, or?
So we start originally with an acetyl-CoA that we then convert to a malonyl-CoA.
Okay, cool.
Yeah, and like so, like this whole process, so this is why we get our carbons from the acetyl-CoA and from carbon dioxide and from those other sources, but like we start with it, but then we turn it into other stuff in order to fit it into the puzzle pieces to throw it into the elongation of the actual fatty acid. And I'm sorry, like this kind of stuff, like there's a lot here, and unfortunately, I don't have this down pat as much as I do like a lot of the Biochem 1 type reactions, because to be completely blunt, get through this stuff because there's like a 2% chance that you'll ever see it again once you get through here. So, like,
All right, thank you.
yeah. So it's get through it enough for the test, and then, it is what it is. But yeah.
All right, sorry for derailing us.
No, you're good. You're good. So malinate is then used for fatty acid synthesis. The final product, fatty acid, will have radioactive carbon in. And honestly, like this, I could see him throwing this as a question. This really doesn't get talked about a ton. This is really more just like understanding the whole process and the whole steps. So if you're using the carbon dioxide can be converted into part of the with bicarbonate. Carbon dioxide and bicarbonate work together a lot. If you remember talking in Biochem 1, we talked about the bicarbonate buffer a little bit. So you can go from CO2 to bicarbonate kind of back and forth.
Yeah, I remember that's where we're pulling a lot of our carbons from.
Yeah.
Okay.
And then we said unlabeled acetyl-CoA, unlabeled malinate. So the malinate is going to be the very, very last thing that's going to get added. And so it's they call it the omega carbon, the furthest from C1, because acetyl-CoA is going to be our first carbon. The CO2 or the bicarbonate will be our carbon, some of the carbons after that. So the malinate will be the very last one. It's just understanding, again, acetyl-CoA is the first one. So if this was labeled acetyl-CoA, then depending if it was just one acetyl-CoA, the answer would then have to be like, like that first carbon is going to be labeled, because acetyl-CoA is where we get our first carbon from when we synthesize everything.
All right, thank you.
I think this is the exact same question, just repeated. Nope, slightly different. This is if malonyl-CoA is synthesized from 14CO2.
Well, we were just talking about the omega carbon. Is that still true for this one?
That's the same question, right? Yeah,
Yep.
That's the exact same question.
Yeah, I feel like he just like switched around some words.
No, I think it's the exact same question. If malonyl-CoA is synthesized from 14CO2 and unlabeled acetyl-CoA, that's the same, and the labeled malinate is then used for fatty acid synthesis, the final product will have radioactive carbon in blank, and then it says no part of the molecule.
Oh, what? All right. Well, we can dispute it, if we need to.
Yeah. If that question comes up, my gut says I like this one better. In my gut. But that's one to, if somebody has a meeting with him and ask him about that to explain it deeper, go for it. That's a lot of blah. Okay. In the biosynthesis of fatty acids, malonyl-CoA is formed. Right, kind of that first step that we looked at, we go from acetyl-CoA to malonyl-CoA. Which of the following pair of compounds are used as the substrates for this enzyme-catalyzed biosynthesis of malonyl-CoA?
And you had
this
just said
one,
bicarbonate and acetyl-CoA?
Yeah.
Yeah, you just mentioned this one.
Gosh, I hate this computer so much. There it goes. Bicarbonate and acetyl-CoA. Ugh. Mitochondrial acetyl-CoA must be converted first into which of the following compounds during fatty acid biosynthesis? So we're looking at mitochondrial acetyl-CoA, right? So we're looking, okay, we have to transport it into the cytosol before we can do all of that stuff. Does anybody remember what it gets turned into in order to cross from the mitochondria to the cytosol? I can pull it back up.
Is that citrate?
Yeah, it is citrate. So remember, we go citrate, oxaloacetate, malate. Can go malate can go directly through. Remember, malate aspartate shuttle, similar to that, or it can go to pyruvate and keep going through and come back around. A couple different options there, but yeah, citrate. It has to get turned into citrate so that it can make it through that membrane. Okay, one of the components in the tricarboxylic acid cycle is used as the precursor for the synthesis of fatty acids. This compound is present in the mitochondrial matrix. This compound must be converted to one of the following compounds in order to be delivered into the cytosol. And if I'm not mistaken, this is saying the exact same thing, just a lot more words. The answer again is citrate. It's literally saying one of the components to make fatty acids is in the mitochondrial matrix. Identify the compound made within the mitochondria which is transported in the cytosol for fatty acid biosynthesis. Citrate. Same question as before, just a crap ton more words.
Yeah, so he's definitely going to want us to know about transport of
acetyl-CoA. He'll want you to know about the transport. More than likely, he'll want you to know which ones cross over. So you'll want to remember citrate, that the acetyl-CoA right here, that acetyl-CoA is going to bind on with, he'll probably want you to know acetyl-CoA binds to the oxaloacetate to turn it into citrate. Citrate goes across and then gives up the acetyl-CoA, and then either malate or pyruvate is going to cross back to be reused and recycled. That's more than likely what he'll ask questions specifically on. Okay. Recognize the compound produced commonly during biosynthesis of both cholesterol and ketone bodies. This is more like in the cholesterol unit, which is like the next chunk of questions. It's HMG-CoA. Whenever you see cholesterol type stuff, always remember HMG-CoA 9 times out of 10 is going to be your answer choice if you're talking about cholesterol. Okay. And then like the acetone itself, acetone is a ketone body, right? And then the other two, it was acetoacetone or something like that, or acetate. And then Kristen, you remember the last one, right? The
something butyrate, right?
buto something. It's like hydroxy.
Beta hydroxybutyrate.
Beta hydroxybutyrate. Yeah. Nice.
Beta hydroxybutyrate as our other ketone body. Okay. Synthesis of both glycerophospholipids and triacylglycerol involves, and we kind of looked at this in a previous question before, that diacylglycerol 3-phosphate. I think we called it something different. It was what did they call it? Something.
Was that the diacylglyceride one?
Yeah, diglyceride. Remember, ide and eights is essentially the same thing. Just like chemically, it's slightly transitioned. So like if we look at like this, if you see It's kind of like if we look at like the like aspartic acid versus aspartate, it's saying the same thing, just a different way to say it. So just like this diacylglycerol 3-phosphate is not like the exact same thing, but just like another step in that process of the CDP or CPD di whatever. Like it's saying the same thing. So when in doubt, guess what's closest. So like you have like CDP choline, but the choline is not, the diacylglycerol is closer to the whole CDP diacyl di whatever. Makes sense. It's not perfect, but honestly, like again at this point, like it's crunch time, so it's, you know, do the best you can to learn as much as you can and remember as much as you can is the important stuff. Okay, the biosynthesis of palmitic acid, it says 16 carbons, requires addition of 2 carbon units in each successive cycle until palmitic acid is made. Which product of the reaction catalyzed by acetyl-CoA carboxylase is this carbon source? So immediately we look, acetyl-CoA carboxylase. What does acetyl-CoA carboxylase do? What's its job? It converts what to what?
It convert acetyl-CoA to the malonyl-CoA?
Yeah, correct. So it's the product catalyzed by acetyl-CoA carboxylase. It's got to be, it makes malonyl-CoA because that's what was said in the book earlier, and that's our answer. Just sometimes a lot of times we like to overthink these kind of questions, but in reality, cheese, highlight, thank you. We like to overthink them, but a lot of times, sometimes if you just try and simplify it, it's going to be a little bit easier. Okay. The biosynthesis of triacylglycerols from acetate occurs mainly in humans with low carbohydrate intake, animals but not in plants, humans after ingestion of excess carbohydrates, or plants but not in animals.
I would say C.
Animals, but not in plants.
Oh.
So actually, if we think about this, olive oil, think about healthy fats, right? Plants as well have fats in them. So a great example, any of our oils, avocado oil, canola oil, seed oils, anything that's in oil based off of a plant, peanut butter as well, all of that fat inside of there is from the natural plants. So plants do produce triacylglycerols. A lot of times it's just they're not producing, like the saturated fats. They're producing a lot more of the mono and the poly unsaturated fats. So it happens in both plants and in animals. So producing triacylglycerols, humans after ingestion of excess carbohydrate is going to be when we are going to have the biosynthesis of the triacylglycerols. Does that make sense?
Kinda. Because there was a question I did earlier, but the answer was animals, but not in plants. Somewhere, I just don't remember where it was. It might have been one of our quiz questions. I don't remember.
Yeah. The thing about this question for me is I don't know where acetate kind of fits into it all, but I just kind of memorized this one. I don't know where I read it though.
So acetate is formed inside of the body under starvation conditions. My guess, just from like a quick look at it, B and C might both be acceptable answers. Let me see real quick. Do plants produce acetate? It looks like some plants can use acetate. I don't know if they... I'm going to say letter C is your best choice. If C is not there, then pick B. Okay? I think C is going to be the best answer for this question. Sorry, I don't have a better answer for you guys. I don't know the full breakdown of all of that from plants and animals. So acetate, from what I can find just as a quick search, acetate can be metabolized and broken down by plants to be used as a food source. And then the human body produces acetate in times of starvation. So, which, again, I don't feel like throughout the book and through a whole bunch of things that ever gets brought up. So kind of, take it with a grain of salt. Okay. The rate limiting step in fatty acid biosynthesis is which of the following? We talked about this super briefly. I highlighted it. It's going to be the
B?
reaction. Yeah, it's B. Our acetyl-CoA carboxylase is our rate limiting step.
And that's the formation of malonyl CoA happens during that step.
Exactly.
Okay.
With using which coenzyme?
FAD8? FAD plus? No. NADPH. Shit. I don't remember. Gosh.
Biotin.
Biotin. Okay.
We
All
use
right,
ATP.
biotin.
We use biotin. CO2 goes in there as well.
All right.
What are the substrates for the enzyme beta-keto acyl ACP synthase? Gross. This is going to be one of the specific ones within the steps. So if we go back to our step-by-step process.
Tell everything.
What'd you say?
Dumbass. I'll admit when I don't know shit and I'll fucking guess. I don't care. And if I'm wrong, I'm wrong. But you don't have to add your 2 cents after I do it.
Substrates for the enzyme beta-keto acyl ACP synthase. Let me look. It's going to be in here somewhere. The beta-keto acyl ACP synthase. Beta-acyl keto. Beta-keto acyl. Geez, stop doing that up there. It's going to be in here. Beta-acyl. There we go. Reduction of the beta-keto group from ADPH to form a hydroxyl group. The beta-keto acyl ACP reductase doing its job in there. Synthase. Jeez. Like I said, there's so many enzymes in here. You guys, I'm sorry.
It's okay. It might be an analog or something.
Yeah. The substrates, we have the acetyl-CoA and the amyl-CoA. So it's going to be one of those beginning steps.
Yeah, it feels like a whole bunch of the steps are just like the process of juggling around the molecules until they can smash them together. It seems like.
Pretty much. That's the whole.
So, with this process and this chunk of stuff, is it safe, especially with as close as the exam is it safe to say that if you don't know, it's either the malonyl-CoA or the acetyl-CoA?
I like this strategy.
That's the way that I would go.
Okay.
It seems like majority of the answers leans that way. So, and that's smart. That's the guessing test taking strategy, which a lot of times I know just kind of the general vibe that I've seen, like even like chats and stuff, like first period of exams, especially in Tri 2, is a really stressful time. So like if you're gonna have exams like this where you go in and you're like, it is what it is. And so it's mostly about like the goal of the whole program, ideally, it's get good grades, learn the material, and like feel good and everything. But at the end of the day, the goal is to get to the next trimester. Okay? So if we can get that, that's a win, because at the end of the day, if you think about it, even like if you have a scholarship or anything going on, like if you lose your scholarship, losing your scholarship is still like at the end of the day, it's still more expensive. Like it's going to be more expensive to repeat a trimester than it is to keep your scholarship. Like ideally, if you can keep your scholarship all the way through, great. But like if you lose your scholarship, it's better to just pass the course than it is to repeat a whole trimester. Like you're in the same boat. So
what I'm trying to tell myself with this try so far is just like, you're still passing, so it's fine.
Try 2 is a doozy, and like I'll be honest, this is where the kind of the part in grad school where these are the weed out trimesters. Try 2 is a big weed out one where it's designed to punch you in the face. And if you can get through this trimester, I'm not going to say it's smooth sailing, but you'll be in a much better position to continue. So, little pick me up there. Okay, question 22. What could be the possible effect of hyperinsulinemia on cholesterol biosynthesis? For some reason, a handful of like cholesterol stuff snuck into this part, but whatever. Hyperinsulinemia, what does that mean?
Yeah, I was going to ask if you could clarify on that one.
Sounds like it's like too much insulin in your blood.
Exactly. Anytime you see hyper means too much. Emia, anytime that you see emia, you're talking about the blood. So hyper, too much, insulin in the blood. Lock those away. Hyper, hypo, you'll see those all the time.
So if you had a lot of blood glucose and a lot of therefore insulin trying to move it out of the blood, would you be promoting cholesterol biosynthesis? Yeah,
exactly. So if you had a whole bunch of insulin, you're going to be looking to create more cholesterol, because cholesterol at the end of the day is fat. It's chains like balls and chains and stuff of fat. So you're going to promote making of cholesterol if you have excessive amounts of insulin. And you think about like kind of the kind of switching gears a little bit more, like the physiology side of things. When you think about specifically like type 2 diabetes, the issue with type 2 diabetes isn't that you aren't producing insulin, because you are. The issue is that you're having to produce so much insulin that your cells actually get desensitized to it. It's like it's constantly being bombarded with insulin. So the cell just kind of like shrinks away, is like, okay, we're kind of done with this. So the solution is literally to then inject yourself with even more insulin to get it to do its job. So, and if we look at the people who are in that group of those, who have type 2 diabetes, tend to also be overweight. They tend to have high cholesterol, have heart issues, have all that kind of stuff. And this is one of those reasons. When we have consistent hyperinsulinemia, it promotes building fat, building cholesterol, doing all those kind of things. I'm going a little bit off track there, but kind of the background of why that is what it is. What is the effect of citrate on fatty acid biosynthesis? So remember, if we have a whole bunch of citrate, is that going to make us want to produce fatty acids, or is that going to make us want to stop producing fatty acids?
Is it an allosteric or I thought it was an allosteric inhibitor.
It's going to be an allosteric activator, because just like we said when we look again like this whole process, if we have a bunch of citrate, excess citrate especially, and the citrate's not being used in the Krebs cycle, it's going to be able to pass through. If we have more citrate available, it's important to remember with all of these things that are happening, these different processes, that they're happening in the body, like they're regulated, but a lot of this stuff is just kind of controlled by things running into other things. Remember when we talked last in Biochem 1 about entropy and enthalpy?
Yeah, the delta G stuff.
Yeah, entropy is kind of the chaos, and like if there's more stuff floating around, there's more likely things are going to collide to be able to do their thing. That's like a lot of this is still in that same idea, where like if we have a bunch more citrate, it's more likely the citrate is going to run into that citrate transporter and get pushed across. So like that's still like kind of the basis of what's happening. Like it's not like we have extra citrate, so our cell goes, oh, we have extra citrate, let me grab you and throw you over there. It's more kind of a little bit more chaotic, a little bit more controlled chaos, if you will. Hopefully that helps just a little bit kind of in the brain understand about that. So if there's lots of extra citrate, the citrates are more likely to travel from the inside of the mitochondria to the cytosol, where it'll then, stimulate, giving off the acetyl-CoA to produce then fatty acids, fatty acid chains. What is the enzymatic function of beta-ketoacyl synthase? Again, if we ever have to guess, acetyl-CoA and malonyl-CoA. Please, come on. Remember, if we ever have to guess acetyl-CoA, malonyl-CoA. And this goes back, watch through and look through the process of the step-by-step. The book here has a lot of words for this. I promise you, if you read through it, break it down into less words, and it'll be much, much simpler to understand. What is the function of the trans-carboxylase enzyme domain present in acetyl-CoA carboxylase enzyme? Acetyl-CoA carboxylase enzyme complex. That's a lot of words. Literally, to just say the acetyl-CoA carboxylase, you can ignore all of this BS crap in the fatty acid biosynthesis. So there's a couple different chunks of that enzyme, which we see right here. So this is just kind of a deeper look at the acetyl-CoA carboxylase, where we have a trans-carboxylase, we have the biotin carrier protein, and then the biotin carboxylase right there. So we look at our answer choices. the function of the trans-carboxylase enzyme domain in the acetyl-CoA carboxylase and the fatty acid biosynthesis. It is actually going to be, because remember, it won't produce acetyl-CoA. Love our guessing. It's actually going to be what produces our malonyl-CoA. See our trans-carboxylase right over here. So it binds in to here, and then acetyl-CoA pops it over to here and makes it into malonyl-CoA. Our trans-carboxylase transfers the activated CO2 from biotin to acetyl-CoA and produces the malonyl-CoA. So again, 50-50 shot if you guess between malonyl and acetyl-CoA. I like our odds as we continue to go through these questions as far as guessing goes.
Yeah, I'm a fan of it so far.
Okay, what type of reaction combines malonyl-CoA and acetyl-CoA to produce the beta ketoacyl ACP, also known as acetoacetylic ACP, on the fatty acid synthase multi-enzyme complex? So if we think back for a sec, we had, right, priming. It was PAC RDR. PAC writer. I don't know, however you want to remember it. So immediately looking here, we're not going to have oxidation. So immediately we have between three different choices. And so this is just going to be looking back at the step-by-step. The question is asking combining malonyl and acetyl-CoA. And actually we can logic our way through this a little bit, right? When we produce malonyl-CoA, that's early, very, very early in the whole process. So one of our first actual processes is condensation. I say this kind of looking at it after knowing the answer. Condensation is our answer, but then actually looking at the step, the condensation reaction, condensation of the decarboxylated product with acetyl group. That's a whole lot of words. I feel like there was a better picture, something that he had, or better thing that like step by step walked you through this. Let me see if I have something in my. I knew I had something better. Did he still have available this PowerPoint right here? Can you guys see this PowerPoint? Is anybody there?
Yeah, we see it.
You guys, did you recognize this PowerPoint?
Vaguely, but it may be from looking at your stuff, so I don't know.
Because he has in here, this is like the whole synthesis, like step by step of where like all the different enzymes and stuff that are part of the whole synthesis, and it goes step by step. They rearrange, and it comes across. It all changes. So now we have the carbons connecting, and then it unfolds again. So kind of the first reduction. If you got my notes, this is in there, and you can kind of use this to kind of walk through the steps. Okay, I don't remember if in my last video in my YouTube video. I don't remember if I broke this down and did this whole thing or not, but like it shows like the whole actual like elongation and step by step, and gives you like what each of these are. Sorry, I just wanted to add, just throwing that in there. What were we talking about? The condensation reaction produced the beta ketoacyl ACP. And I didn't really see a lot in the book on this. I personally feel like Sarkar's book is really, really good, but I feel like a lot of like this first chunk of material, I felt like a lot of Biochem 2, I feel like there's a lot of background that genuinely felt like there was some background that I was probably missing when I took the class, but it is what it is. Okay, which enzyme uses malonyl-CoA as its substrate for fatty acid chain elongation? We're just going to run through these. The endoplasmic reticular elongase. And let me actually... Why he picked that color? Reductase, transferase, dehydratase. I don't even see that in here. I swear half the stuff isn't even in the book. I believe in you guys. Which of the following biochemical reactions potentially inactivates the enzyme HMG-CoA reductase? Whenever we see HMG-CoA reductase or HMG-CoA, what did I tell you guys to think about when you see HMG-CoA?
Cholesterol?
Yeah, think about cholesterol. So, inactivating the enzyme HMG-CoA reductase, it's going to be phosphorylation. There is what is going to inactivate it. And that's just kind of a memorization thing. Phosphorylation sometimes activates, sometimes inactivates. HMG-CoA reductase gets inactivated with phosphorylation. Which of the following compounds is required as the reducing agent during fatty acid biosynthesis? This is going to be our NADPH. Remember, it's going to be this because reducing agent. So do you remember what the difference between a reducing agent and an oxidizing agent is? Which does which?
So the reducing agent needs to be oxidized and the oxidizing agent needs to be reduced.
Yeah, so the reducing agent gets oxidized, so it gives away electrons. It gives away a hydrogen. So it's a reducing agent gets oxidized. The oxidizing agent gets reduced. So like in dehydrogenase reactions, those we have oxidizing agents. Whereas in synthesis reactions, we're having reducing agents. Okay. Which of the following compounds is required to lengthen the palmitic acid chain by 2 carbon unit within the mitochondria? We have a compound to lengthen the palmitic acid chain by 2 carbon units within the mitochondria. 2 carbons, we look at all the stuff that we have here. Mevalonate, nope, that guy has 3 carbons, so can't be that one. Gosh. 2 free carbons, we never have free carbons. The bicarbonate, it's not going to be that guy. Our age-old guess, acetyl-CoA or malonyl-CoA, still holds true. I love that for us.
Me too.
I'm at least getting a 50.
Who cares about studying? Let's just guess malonyl and acetyl-CoA. Let's see if it still holds true. Which of the following compounds is the donor of 2 carbon units in palmitic acid biosynthesis? We
Acetyl-CoA.
have both of them here. Immediate
Yeah,
50-50
acetyl-CoA.
shot. Answers in this one's going to be malonyl-CoA. Again, because the acetyl-CoA gets turned into the malonyl-CoA in order to go through the steps to then be added in. Which of the following compounds is the principal product of fatty acid biosynthesis and the precursor of other long-chain fatty acids? This is why we talk about it so much. This is palmitic acid. It is the principal product of fatty acid biosynthesis. So we make palmitic acid, and then if we need to continue to make other stuff, that's when like we mess with it and add or take away or add extra stuff or do whatever to form whatever it is that we need. That's another one. If it's palmitic acid, that's another one to guess on. If palmitic acid is in there, like there's a good chance the answer is probably palmitic acid. Which of the following conditions can inhibit fatty acid biosynthesis? Inhibit fatty acid biosynthesis. So we look at this first choice, increase in cytosolic citrate concentration. We already talked about citrate is an allosteric activator. Formation of acetyl-CoA carboxylase polymers. I mean, if we're if we're having those, making those polymers, that means that we're producing fatty acids. Increase in cytosolic palmitic acid concentrations immediately. When I'm looking at that, going to be that one, because palmitic acid is our end goal. Remember, if we have our end goal, it's going to come loop back around and we get negative feedback. So palmitic is our is going to inhibit because we already have our end goal of what we need. Which of the following enzymes is the rate-limiting enzyme for cholesterol biosynthesis? Always guess. HMG-CoA, so that's stuff. So it's our HMG-CoA. Reductase is our... And again, I don't know why this is included in this unit. It should really be included in the cholesterol stuff. Which of the following enzymes could be one of the major important producers of reducing equivalents during fatty acid biosynthesis? So reducing equivalents. So we're talking about stuff getting reduced. If stuff is getting reduced, right, we have those reducing agents. What's our main reducing agent that we're using during fatty acid biosynthesis?
Say that again.
What is the major reducing agent that we're using during fatty acid biosynthesis?
Deuce.
When I say it, it'll be like, oh, yeah.
That's not the NADPH, is it?
It is, yeah, our NADPH. Reducing equivalent is similar to saying reducing agent. Like in this context, it means like the same thing. So this question is basically asking, like, which enzyme produces the reducing agent that we use during fatty acid biosynthesis? And if you remember, there's kind of those two big things. We get the NADPH from that pentose phosphate pathway, and also from, it's on the book, that malic enzyme reaction way up at the top. Right there. Sources of NADPH, that pentose phosphate pathway, which gives us 2, or malate to pyruvate with malic enzyme also gives us an NADPH. So another choice in here as well, another perfectly correct answer choice would be pentose phosphate pathway, or like, yeah, I don't think he'd give you like the specific enzymes of the pentose phosphate pathway.
I feel like he does ask a question about this, that the answer is pentose phosphate pathway, though.
Probably.
I don't know if that was on a quiz or when I was putting these in Quizlet, but I think he does.
Here's one we should know. Which of the following enzymes is activated by the vitamin biotin? Say it louder. You got it.
The acetyl-CoA carboxylase?
Yeah, or acetyl-CoA carboxylase. What does the acetyl-CoA carboxylase do again? What's its job?
It initiates the reaction that changes the acetyl-CoA into the malonyl-CoA.
Good. And what is all used during that reaction? What other extra stuff do we have to use?
ATP and bicarbonate.
We'll call it, yeah, or we'll use bicarbonate or CO2.
CO2.
Whichever one. We typically say bicarbonate.
I don't
And what about the
remember the
coenzyme?
other one.
What about the coenzyme that we use?
I don't remember it.
It's a B vitamin. B7 to be exact. Biotin. Use our biotin. Which of the following enzymes is required to produce mevalonate during cholesterol biosynthesis? See, again, we're in the cholesterol, which is technically the next chapter. But anytime that we see cholesterol, what's our immediate go-to guess?
HMG-CoA.
And our HMG-CoA reductase is going to be one of our rate-limiting steps. So it's super important. So if you see that reductase in general, that's our other good one to guess when it comes to cholesterol and enzymes. Which of the following fatty acids is produced if palmitoyl-CoA is elongated by 2 carbon units?
Is that
So
C?
we look at this. Remember, palmitic acid is going to be how many carbon units?
16.
It has 16. Oleic acid, I'm just going to write these out in here so we have them. So this is a 16 carbon. Oleic acid, these are the ones that we want to remember, is an 18 carbon. Palmitoleic acid. This one is also a 16 carbon monounsaturated. Stearic acid is also 18. And palmitoyl is also going to be 16. So immediately, if we add 2 to it, we have a 50-50 shot, oleic versus stearic acid. In this case, the answer is going to be stearic. Unfortunately, this is more just kind of like know it and remember it, which sucks, but it is what it is. Okay. Which of the following is a lipid molecule? Diacylglycerol, that's acylglycerol, it doesn't include lipids. Collagen fibers are going to be made up mostly of glucose. Ethanolamine, this is an amine, not a lipid. CDP choline is the only one that can be a lipid molecule. Which of the following is a phospholipid. This again, all of this is going to be going through and looking at that kind of that chart at the end. But also we can do a little bit of guess and check. Glycerol, right here, glycerol is an amino acid. Glucose 6-phosphate, that is a carbohydrate-based molecule. Sphingomyelin, that one is going to have lipid in it. And oleic acid, that's a fatty acid chain. So no phosphorus involved there. So best guess, sphingomyelin. And that is correct. So you can either, practice these questions enough. It's like kind of these easy give me's, like click and move on, or, kind of work your way through it a little bit. Which of the following is not a site of fatty acid synthesis? So we look through all of these. Adipose tissue, 100%, because that's fat tissue. Liver, we know, always 100%. Mammary glands. Mammary glands are super. We need to talk about like mammary glands and stuff. This is super, super, super cool stuff happens with mammary glands. Like so much crazy, crazy stuff happens. And they do produce fats because they need to produce fats in order to put fats into the milk when a mother is breastfeeding. So our answer is skeletal muscle. Also, if you think about dissection, come on, computer. If you think back to your dissections, once you actually get to the actual muscles themselves, like inside of the muscles doesn't have fat. All around, maybe like in between muscles and stuff, you'll find lots of like fat adipose tissue, but actually inside of the muscles themselves is just pure muscle tissue. Kristen, did you have a comment or question?
Sorry, I was typing. That was my bad. Try to turn off my.
No, you're good. Which of the following is not required in the biosynthesis of fatty acids? This one we should know. Of all these things, which one are we not going to use?
NAD+.
Perfect. Which of the following is not true about synthesis of malonyl-CoA during fatty acid biosynthesis? It requires carbon dioxide or bicarbonate ion. It requires biotin. It requires activation of acetyl-CoA carboxylase. It requires acyl carrier protein.
D.
D.
What'd you say? Sorry.
D.
Good, D. Because we do need the biotin. This is one of that very first steps. Because remember, acyl carrier protein is further down in the process. Which of the following is not true of the reaction forming malonyl-CoA during fatty acid biosynthesis? As you can tell, he likes that first step. There's going to be a lot of questions on kind of that first chunk. The reaction forming malonyl-CoA during fatty acid biosynthesis. I think this is almost the exact same question, where it requires
Same answer.
same answer, just a couple different answer choices. Which of the following is used to activate phosphatidic acid for conversion to various phospholipids? The answer is CTP. I don't remember exactly where this is, but hopefully an easy enough question to remember if it comes test time. If you see all these, pick CTP. Because these other ones, UTP, TTP, and GTP, aren't ones that we discuss this trimester. That was last trimester. Oh, here we go. List of the enzymes required to transport a molecule of acetyl-CoA from the mitochondria to the cytosol while returning the acetyl-CoA acceptor back to the mitochondria. So we look all through this. And this, honestly, this question is just really a good one. Rather than like drawing out that whole process, use this question as a guide to learn that. Citrate synthase, citrate lyase, malate dehydrogenase, malic enzyme. So this one right here. So if you can remember CCMM, good for that whole process. The more you can do to just, the more ways you can have little tiny reminders, the better off you're going to be. Question 47. Which of the following moiety in the fatty acid synthase complex is directly associated with the vitamin B5 containing a cysteine sulfhydryl group? This one is going to be, I'm just going to give you guys the last of these answers so we start to recognize things and we can keep working, so you can keep studying stuff. So that's an acyl carrier protein. So then you know if you ever see sulfhydryl or like sulfur containing, think acyl carrier protein. Make that connection in your brain. Which of the following molecules provides energy for the formation of carboxybiotin in fatty acid? Provides energy. Immediately provides energy. What is our enzyme, or what is our molecule that's whose job is it to provide energy?
ATP.
Always ATP. It's ATP's job to provide energy. Which of the following proteins is an integral component of the enzyme fatty acid synthase multi-enzyme complex? So real quick, it says protein of the enzyme fatty acid synthase multi-enzyme complex. So this one is going to be, again, the acyl carrier protein. So as we get down here towards the bottom of stuff, this is another thing I'd add onto your list of look through the book for acyl carrier protein stuff and focus on understanding kind of what that's about, because it does seem like he likes to focus on acyl carrier protein. Which of the following statements about the beta oxidation of fatty acids is true? I'm back a chapter for some reason. Cool. 2 NADH are produced per acetyl-CoA. That is wrong. Why is this wrong? Why is it 2 NADH are produced per acetyl-CoA? Why is that wrong? Because it's not 2 NADH, right? It's 1 NADH and 1 FADH that we produce. See, 2 moles of acetyl-CoA are produced per mole of palmitic acid oxidized. And that's not true because palmitic acid, right, has 16 carbons. So how many moles of acetyl-CoA would we produce from
8.
a mole of palmitic acid? Exactly, we produce 8. About 1,200 ATP molecules are generated from the beta oxidation of a 20-carbon fatty acid. Typically, he won't ask you like specific numbers, that kind of stuff, so meh. The free fatty acids with more than 14 carbon atoms must be converted to their corresponding thioester before the process of beta oxidation commences. That is going to be our answer choice. Also, kind of, if we think about what else about 14 or more carbon atoms is important. Remember, if we think back about the transport of those atoms, if we need, and that's just to say molecule, not atoms, 14 carbon molecule. If we're more than 14 carbons long, we need something to help us, right? Because we need to convert it to a thioester. So like attaching on, making it an acyl-CoA in order for it to pass through that mitochondrial membranes. Do you remember back about that in beta oxidation, where if it's
Yeah,
more
it was
than
choline that it needs, right?
Yeah, choline is part of it. Yeah, no, it was the two. It's the transport. It's the carnitine.
Carnitine. That's right.
Carnitine.
I knew
So
it was a
beside us
C.
all, and it turns into an acyl-CoA so that it can go with the carnitine acyltransferase 1 and then carnitine acyltransferase 2 to get into the mitochondrial matrix if it's more than 14 carbons long. Because more than 14 carbons long is just too big for it to pass through uninterrupted. Which reaction activates beta-hydroxymethylglutaril-CoA or HMG-CoA? It's so stupid. Just know the full name. That's the full name of HMG-CoA reductase. So what reaction activates it? We mentioned this before. How remember phosphorylation inactivates it. So dephosphorylation is going to activate it. Phosphorylation inactivates it. Dephosphorylation activates it. Which statement is not true about precursors required for fatty acid synthesis in animal cells? NADPH is produced in the nucleus by malic enzyme. There's a lot of words here, but the answer is this first one because the NADPH is not produced in the nucleus, right? It's produced in cytosol with that pentose phosphate pathway, with that kind of stuff. And then eventually, like that, if we do need, remember the purpose of the pentose phosphate pathway is to make the ribose, the ribulose, xylose, that kind of stuff. And then if we need to be making DNA, then the ribose goes into the nucleus and does its thing.
Yeah.
So, but it's not produced in the nucleus. Jeez, that was a long 27 pages. Thank you guys for sticking with it. There at the end, like once we got through a lot of the yuckiness and kind of figured some stuff out, I feel like, especially towards the end, it was a little bit easier to crank through them, and more of them seemed a little bit more familiar. Hopefully you guys felt the same way.
Yeah,
I
for
will
sure.
say this, I just looked at 5 and 6, and they just keep getting longer from there.
Yeah, there's a lot of information in 5 and 6. So what I'll say, no matter what happens, if this first exam does not go the way you want it to go, it's kind of it's gonna serve as a reminder that even though Try 2 is very, very, very busy, this can't be a back burner class. So, and we'll keep up with it. We'll try and be more consistent with tutorings right up from like preparing all for exam 2 as well. So we'll have a little bit more time to prepare than we did for this first exam. Okay?
Thank you so much.
Thank you, Drake.
Thank you, sir.
Okay, guys. Appreciate you all for being here. Have a great night. Take care. Get some sleep.
Go enjoy
you
North
too.
Carolina.
Yeah, have fun.
Yep.
Will
Have
do.
fun.
All right.
Good night, you guys.
Bye.
Good night.
Thank you, Drake. Bye.